In the 1950s and 60s, neuroscientists David Hubel and Torsten Wiesel asked a fundamental question: how does our brain process vision? Their groundbreaking experiments with cats revolutionized neuroscience and won them the 1981 Nobel Prize. Today, we'll explore how they discovered the secrets of how we see the world. Their experimental setup was ingenious yet simple. They anesthetized a cat to ensure it felt no pain, then positioned it comfortably facing a projection screen. Using hair-thin microelectrodes, they carefully inserted recording devices into the cat's visual cortex. These electrodes could detect the electrical signals when individual neurons fired. Connected to recording equipment, they could now monitor the brain's response to visual stimuli in real time. Now came the experiment itself. They projected various light patterns onto the screen while carefully monitoring neuron responses. At first, random spots of light produced minimal neural activity. But then came the breakthrough. When a line or edge moved across the screen, suddenly there was a burst of neural spikes. Zooming into the brain, they could see specific neurons lighting up. They began testing different orientations. A vertical line caused specific neurons to fire. A horizontal line activated different neurons. A diagonal line triggered yet another set. As they rotated the line through 360 degrees, different neurons activated sequentially. The discovery was remarkable. Individual neurons responded to lines at specific angles, one neuron for vertical, another for horizontal, and others for every angle in between. Hubel and Wiesel identified three distinct types of cells in the visual cortex. Simple cells respond to lines at specific orientations in specific locations. Complex cells respond to moving lines regardless of their position on the screen, and hypercomplex cells respond to lines of specific lengths or to corners and angles. Each cell type plays a unique role in building our visual perception, working together in a hierarchical system. But Hubel and Wiesel discovered something even more crucial. When they raised kittens in complete darkness, or in environments where they could only see horizontal or vertical lines, these kittens developed abnormal vision. Their neural connections formed improperly. This revealed what we now call the critical period, a window in early development when proper visual input is absolutely essential for normal vision to develop. This principle applies to human babies as well, which is why early detection and treatment of vision problems in children is so important. So what did Hubel and Wiesel prove? First, that vision is hierarchical. Simple features like edges are detected first, then combined into increasingly complex objects. Second, the brain has specialized feature detectors for different visual elements, from lines and angles to movement and depth. Third, early visual experience during the critical period is essential for normal development. And fourth, the brain doesn't work like a camera simply recording images. Instead, it actively constructs our visual experience by detecting and combining features. Their work laid the foundation for modern neuroscience, influenced treatments for childhood vision disorders, and even inspired artificial intelligence and neural networks. Thanks to Hubel and Wiesel, we understand that